So, all of Hunter's workshops have been like hardware, chemistry, physics, but today we're going to touch on some biology. So, unfortunately, you can see that stuff up there, but today we're going to go over some chemistry terms, and then we're going to look at some case studies from IBM, Google, and Rigetti Computing, and then we're going to look at some quantum in biology, like in nature. So, the background. The first term that you should know is Hamiltonian, which is just the energy operator of a molecule comprised of kinetic plus potential energies, or T plus V. The ansatz, it contains adjustable parameters which control the quantum state the circuit prepares, and it's German for starting assumption. Coherence is just the phase relationship between the quantum states that enables interference. QPE, or quantum phase estimation, is a quantum algorithm used to determine the eigenvalue or phase associated with an eigenvector of a unitary operator. It works by applying controlled powers of the unitary operation and using the quantum Fourier transformation to extract the phase encoded in the quantum state. It can be more precise, but it's harder on noisy hardware. And the last one is the quantum Fourier transformation, which is just the quantum version of the classical Fourier transformation, which is just used to extract frequency or phase information from a quantum state that would otherwise be hidden. And then this is also a term, but it needs its own slide, it's very important. So VQE, or variational quantum eigensolver, is a hybrid quantum classical algorithm used to estimate the ground state energy of quantum systems, especially molecules. The molecule's Hamiltonian, which is T plus V, is mapped to qubits, so it can be represented as a quantum circuit. So looking at this diagram here, we start up at the top where the squid is. So the squid represents the ansatz because the squid has many arms, just like the ansatz can control many parameters at once. And then we go down to the circuit, which is the quantum circuit. So we run it on a quantum computer, and it produces a quantum state, Q sub F, which is our current guess for the molecule's ground state. And then it goes over to the bank. So the quantum computer measures the values of the Hamiltonian, the H target, to compute the energy. This energy acts as a loss function, which is what we want to minimize, which is why it's a bank. And then lastly, we just repeat the cycle until convergence, which is when the measured energy and circuit parameters, or theta, is the lowest energy possible. And and it gives us an approximation of the ground state energy. So here's just like a simulation showing what that cycle is doing. So it keeps measuring at different parts, and then it keeps trying to find the lowest and lowest point, and it will keep measuring it until it finds what it thinks to be the lowest point. And it should get to it soon. There. But, yes. And then we have VQE, which is what I just showed you, versus QPE. So VQE, its pros are that it works on noisy hardware. NISQ stands for Noisy Intermediate Sub-Quantum Hardware. And it also has shallow circuits, and it has flexible ansatz choices, again, the squid. Its cons are that optimization can get stuck. So I'll show you here. If we're here, for example, and we try to optimize it, it will think that this is the lowest point because it's not doing a very deep search. So it thinks that right here is the lowest point when it's not actually the lowest point. And other cons are that accuracy depends on the ansatz, and many measurements are needed. QPE, on the other hand, it's more deep. Its pros are that it has a higher theoretical precision, direct eigenvalue estimation, and no variational optimization. The cons are that it's deep, so it's noise sensitive, it requires long coherence times, and harder on current hardware. So now let's look at some case studies. So the first one, Google. In 2016, researchers at Google used a quantum processor to simulate the hydrogen molecule and compute its energy surface. The goal is to calculate how the energy of the molecule changes as the distance between two atoms changes. The experiment used quantum phase estimation, which can estimate the eigenvalues of a Hamiltonian, and therefore was mapped onto qubits, so the quantum processor could simulate the behavior of the electrons in the molecule. By running the algorithm for different interatomic distances, which is just the distance between the molecules, the researchers were able to calculate the molecule's potential energy difference, energy surface, sorry. The lowest point on the surface, which is shown on the x-axis on the left graph, corresponds to the equilibrium bond length, which is where the molecule is most stable, and the y-axis shows the total energy of the molecule. So on the left graph, that black curve, that's like kind of with the red curve, is the exact theoretical energy calculated using classical quantum chemistry methods, and the red dots are results from variational quantum eigensolver experiments, and the blue points are from the quantum phase estimation experiments run on a quantum processor. So going back to this, Google used both of these. The rest of the experiments will only use VQE. And the right graph shows the error in the energy calculation for different bond lengths. So that straight line across is the chemical accuracy threshold. So we want to keep all of our measurements below that. And as you can see, the red line is below it. And the green dots on the right graph are from the phase estimation, and they show larger errors due to noise and experimental limitations. And the results matched theoretical chemistry calculations, showing that a quantum computer could reproduce known molecular energy values. And this experiment was an early demonstration that quantum computers can simulate molecular systems, which is important for future applications in chemistry, material science, and drug discovery. So here, this is just showing like the different bond lengths. So as it calculated all the different bond lengths and the energy, and then it found like around here, it was the best bond length for it to have the lowest energy. And then because we're dealing with noisy stuff, as you can see, like this is what we want to see, but unfortunately it's very noisy, so this is unfortunately what we do end up seeing. IBM, so I'm just going in chronological order. So in 2017 now, IBM researchers used the variational quantum eigensolver on real superconducting quantum computer to simulate the hydrogen molecule, again using the hydrogen molecule. The goal was to compute the molecule's ground state energy, which determines how stable the molecule is and how it behaves chemically. They mapped the molecule's electronic Hamiltonian, which describes all the electrons in the molecule, onto qubits and used VQE to find the energy. And the graph shows the interatomic distances versus the energy. The curve represents the potential energy surface of the molecule, with the lowest point on the curve being the equilibrium bond length. And the dots are from the quantum computer, and it shows that it closely follows the theoretical curve. So Google's experiment showed that quantum computers can reproduce the energy curve of a molecule using phase estimation, while IBM's experiment showed that hybrid algorithms like VQE could do similar chemistry simulations more effectively on noisy hardware. And this was one of the first demonstrations that showed that quantum computers can perform real quantum chemistry calculations even with only a few qubits. And if you've been to any of Hunter's workshops, he talks about how we don't have enough qubits. So this is showing that even though we have very few qubits, we can still do some cool stuff. And then lastly, Rigetti Computing. They just developed superconducting quantum integrated circuits for quantum computers, and they use their own circuits for their own case study. So they used a variational quantum eigensolver to compute molecular ground state energies, and they tested molecules like sodium hydride, potassium hydride, and rubidium hydride on their Rigetti Computing superconducting quantum processor. And the reason that they did hydrides, there's a few reasons actually. So the first is that it has relatively few electrons, making the simulation small enough to run with limited qubits. But on the other hand, they're chemically ballistic molecules because they have enough, they're not simple enough molecules like hydrogen was that would make it like too easy. It's showing like just a bit of complication. And because they are all hydrides going down the alkali metals, they have increasing complexity, and it's easy to see how this would scale. And this demonstration showed that near-term quantum computers, which are just quantum computers we expect to have in the near future, can perform accurate molecular energy estimation. But again, it's a proof of concept step towards scalable quantum chemistry simulations. Okay, now we're going to look at quantum in nature. So we just looked at what we're using quantum to study nature, but we can actually see in nature that they have quantum stuff that's going So many biological processes depend on molecular structure and energy because at the most fundamental level, biology is driven by chemistry. Chemical bonds determine how atoms form molecules and what shapes these molecules take, which affects biological interactions like protein folding, drug binding, and how biomolecules interact inside cells. Molecular stability is what we just saw examples of. And so vision. Vision begins with a molecule called retinol inside the rhodopsin pigment in the retina of our eye. When a photon hits retinol, it rapidly changes from an 11 cis retinol to an all trans retinol, which is when the double bond rotates, changing the molecule from bent to straight. And then that activates a signaling cascade, which ultimately produces an electrical signal that's sent to the brain where it's interpreted as vision. Enzymes, biological reactions occur along specific energy pathways that pass through transition states, and understanding these pathways explains how enzymes accelerate and how complex biochemical processes proceed. So specifically, chorsmet mutase is an enzyme involved in amino acid synthesis. Quantum chemical simulations of this chorsmet mutase have shown that enzymes stabilize the transition state, lowering the reaction's energy barrier, which is ultimately how enzymes are catalysts. And then photosynthesis. The Fenner-Matthews-Olson complex, or FMO, is a sunlight harvesting system found in green sulfur bacteria living in low light conditions. And experiments suggest that quantum coherence may help the excitation energy explore multiple pathways and reach the reaction state more effectively, which is basically what we do with quantum computers. So understanding how these processes work, especially like the enzymes, is very useful when we're designing drugs. And today's demos from IBM, Google, and Rigetti Computing are small, but they establish the workflow needed for larger biological systems. And that's it.
Cool. May I ask you a few questions after this applause? Can you go back to the quantum in nature slide? Yeah, I think the previous one. So just to kind of clarify a few things. So for the vision one, is the reason that that's kind of considered quantum because it requires very specific energy of photon, and like that's like a quantum physics phenomenon?
Yeah, I'm not sure the specifics of it. There wasn't as much stuff out there online as there was for enzymes. But I think what happens is it sees like the most potential, like the most stable bonds that it can create. And because of this like cascade that it does, I'm not sure, it just like, it signals our brain that we have something going on, and it finds the fastest way possible to see what we're actually seeing.
Okay, interesting. So is that kind of like because of the photosynthesis, it's almost like an optimization problem where it's trying to like reach the receptor areas as fast as possible. So is it almost like that where it's like an optimization?
Yeah.
Interesting. Yeah. And then just one more question as well. So like you talked about like the modeling of molecules with quantum. Mm-hmm. Do we know what the limits of classical computing are? Like can you obviously like quantum is better than classical, but what are the limits with today's technology?
Basically just time. Because we have the ability to calculate all of like the different bond lengths, but in order to find the best, we have to find all of them and then find the best. So using a quantum computer is faster.
Right.
Whereas we can't do it on a classical computer. It just would take forever.
Right. And I guess like the dimensionality, I know a lot of like medical molecules are like extraordinarily large. So I guess like the dimensionality could like exceed what's realistic.
Yeah.
Pretty fast.
And in terms of like drug discovery, just like in material science, how they use quantum to basically like simulate different materials before actually building them, because it takes forever to build a material. They would do the same with drugs, where they would use a quantum computer to do what they have been doing with classical computers, and just testing molecules in different situations, seeing how they bind to each other and in our body, and then go ahead and actually design the drug like in real life. Just makes it faster.
All right, interesting. All right, thanks a lot, Emily.
Any other questions?
All right.